All right, everyone, let's jump into chapter 14, where we're going to be talking about the non-specific immune response. What I mean by this is when we get into the non-specific immune response, we're talking about your body just reacting to, bad guy. But whenever we get into the specific immune response, we are reacting to a very specific thing, and usually even a specific part of like a microbe or something like that. So we'll get into this. Okay, before I forget. So in this chapter, we're going to talk about the role of the first line of defense in the immune system. The first line of defense is going to be like your skin and stuff like that. So it's not even really necessarily part of an immune system, but it is important it does play a role in protecting your body as part of the immune system. So we're going to talk about systems that are involved in detecting infection, the functions of blood cells in the immune system, and the reaction of the immune system to tissue injury. And that's going to be talking about inflammation and the point of it and how it's any good. So essentially, here are some of our defense mechanisms of our, our body. I say our body, our immune defenses. Okay. We're going to start talking today about this is your first line of defense, and this is your second line of defense. You may be like, it says that on there, but like, I definitely want you guys to be aware of what is part of the first line, what is part of the second line, and what is like innate versus what is acquired or specific. That's the same thing. So that's the idea there. The first line of defense is usually typically the physical and the chemical barriers. This is going to prevent any entry at all. Right. So the second line of defense is pretty much mostly what we're going to be talking about in this chapter, but that's going to be once a thing gets in, how are we going to resist it? Okay. Later on in the next chapter, we're going to be talking about the acquired or specific immune response, and that is going to be eliminating. That's using like antibodies and cells that kind of respond to the antibodies or make the antibodies or help trigger making the antibodies. A lot of stuff like that, and to target specific bad guys quickly to get rid of them, especially forming memories. So that's where we're going to see that later. But so where in our bodies would we be exposed to any sort of invasion by any invaders? We have our skin. Obviously, we know that our skin is most of our body is covered in skin. So that's something we want to be able to make sure that it's safe and it's protected. Then we have our mucous membranes. Our mucous membranes are going to be all over our GI tract, our respiratory tract, our genital tract, and our urinary tract. And our eyes are kind of also exposed as well. We consider them part of the mucous membranes. You could in a way because of like the moisture area and like up underneath like the eyelids and stuff, those are sort of like mucous membranes, the undersides of your eyelids and things. But the eyes themselves wouldn't be mucous membranes, but they are still exposed, right? So I mean, you can close your eyes, but like, what if you don't have your eyes closed? So that's another area you could be exposed. You might want to have protection from. So this is a chart sort of looking over the different areas of the body and where we might have lines of defense in place already. In our skin, we have our sebaceous glands producing oil. So oil makes it hard for things to attach. In our tears, we have something called lysozyme. If it has zyme, then it's an enzyme. It's going to break things down. Lysozyme is going to break down other molecules and break down invaders' molecules as well. That's in our tears. And we have mucus. Mucus gets sticky. Mucus helps, trap things. And we have saliva, which also has lysozyme. In our respiratory system, we have cilia, which move, they're sort of like appendages or whatever in cells to help move mucus out. So moving microbes that might be stuck in mucus out. So that also includes the mucus here. Our stomach acid is low pH. It'll kill things. We have intestinal enzymes that break food down. That can be deadly to things. They also have mucus in there. Pooping just pushes things out altogether. And then we have urination that'll flush out stuff as well. Intact skin is just like a literal actual barrier that you cannot get through. Like it's actually like just, it's a barrier physically. Then we have earwax to trap microbes and to help move microbes out away from us or any other debris. Low pH on the skin is going to be harmful to bacteria, not growth friendly. And then we have sweat with, the sweat itself might have enzymes in it, as well as salt and a bacteria unfriendly pH and stuff like that. So that's sort of our first line of defense breakdown there. So we're gonna go through these. Skin, we have the epithelial cells, which are our skin cells that are really closely compacted together and even cemented together by molecules that are outside of the cells, part of the extracellular matrix. And they're impregnated with keratin, and keratin is essentially a waterproof protein that helps kind of seal it all up. So we'll say that. So that makes a nice, thick, tough layer that is impervious and waterproof, and not very many pathogens can penetrate that. Hair follicles themselves, we have a hair shaft that is periodically extruded, so we get rid of it periodically. And then the cells of the follicle will themselves be desquamated, and that just means shed regularly and replaced. So that helps prevent microbes from really taking up residence there. Sweat glands also have a flushing effect. The glands themselves do, and the sweat itself isn't usually very microbe friendly. Mucous membranes in general are moist. They are permeable, but this is a problem, right? But they often have a flushing effect that can be replenished, especially if you are, well hydrated and you're taking care of your body, which most people aren't. They also provide barrier protection, but they don't have a keratinized layer. It's just a barrier, physically. They have a mucus coat to impede entry. The mucus coat itself keeps the bacteria from getting in, and then also the mucus coat can help with flushing and pushing away any microbes that are trying to get in. So that helps deal with that permeable sort of aspect of it. The respiratory tract has nasal hairs to trap things. Then we have copious flow of mucus, obviously, throughout your sinuses, through your throat and everything, and even in your lungs themselves. And then we can have movement of that based on the ciliated epithelium that is going to trap the mucus and move everything out of the lungs towards the pharynx for removal, like coughing stuff up. So here are some non-specific chemical defenses, chemicals that our body produces that are not specific to any specific microbe, but microbes in general. In our skin, we have sweat with lysozyme, which is that enzyme that breaks things down. It breaks often specifically organic molecules down. Then we have salt, which is not always friendly for cells, and then low pH. Remember why salt is not friendly for cells? Because of the osmotic pressure. Caused by that hypotonic, hypertonic balance, right? And low pH means acid. That's not friendly, of course. This is, I don't know what that is. Probably an Amber Alert. Missing persons, just FYI. I just heard it going off in the whole freaking department. Anyways, that really distracted me. Mucous membranes, they're sticky, they're thick, they have antibodies. Oh, also the oils on the skin, I skipped over that, but obviously they're slick to microbes just like they are to us. So it's, and they also have antimicrobial fatty acids, which themselves can be not bacterial friendly. So yeah, so the stomach itself, low pH with the hydrochloric acid. Intestines have digestive juices and bile. Bile is a little bit higher in pH than what water in the body would be typically. And then we also have other bodily fluids like semen having antimicrobial chemicals in it. And then the vagina itself has a protective acidic pH that is maintained by the normal biota, typically lactobacillus. Anytime you want to move. All right, lysozyme, breaking this down a little bit. It's a enzyme found in tears and saliva. It hydrolyzes specifically peptidoglycan in the cell walls of bacteria. So it's breaking down some organic molecules. That's what we think of it as doing. It can break down some other molecules as well, but this is what we think of whenever we think of why it's important and what it does for our body. It breaks down peptidoglycan. It breaks down the cell walls of bacteria. So they can start falling apart and we can attack them and get rid of them easier. So moving on to the actual immunology portion, the second and the third lines of defense. These are what we're going to be talking about for the rest of this chapter and then the rest of the next chapter. There's a very important concepts here dealing with, recognizing bad guys versus non-bad guys and all that I feel like can be pretty interesting, and I'm hoping that you guys will enjoy it as much as I typically do. So in these lines of defense, we have to be able to recognize that invaders look different compared to our cells. So we're able to recognize self versus non-self. Self is your own tissues and your own surface proteins and everything associated with self, right? We know this as this is, friendly. Anything with this is friendly stuff. Non-self stuff, anything from outside, we want to recognize that as a, this doesn't look right. This is, bad guys stuff. Now, that's how our body's going to look at it, but that's not necessarily always the case for everything, right? If you get an organ transplant, your body can see that new organ as being non-self, and so it will attack it as it was bad. That's what organ rejection is. So we have to kind of break it down and get a little bit specific with this. But at first, with this second line of defense, the innate immunity stuff, we don't really get too specific with it, luckily. But we'll get into that stuff in a minute in chapter 15. What do I want to say about this otherwise? Right. So if we have white cells that are like protecting us, it's the purple guy. Purple guy's coming around and he's interacting with cells in its environment. It sees these guys out here and it says, oh yeah, no, I know you. No, never mind. Bye. And then scoots off onto its own little world. However, when it sees something that it sees as has something on there that it recognizes as foreign, some cells have receptors that interact with specific foreign molecules. So in this case, we have a pathogen recognition receptor that interacts with PAMPs on the outside of a microbe. This is a pathogen associated, I believe it's microbial product or protein. I think it's microbial protein. Anyways, it doesn't really matter. We'll get to it in a little bit. But anyways, it recognizes the bad guy protein, right? It says, yeah, you're definitely bad. It has a trigger in there that allows it to say, no, this is definitely bad guy. Let's go ahead and eat this guy up. And then we're going to bring it inside and break it down into little pieces down here and where it says destruction. So that's the idea of how that works in the innate immune system, which is non-specific. Okay, so in the body, we have different compartments within our body. And microbes don't just stay on the surface. So how do they get deeper into deeper tissues? How do they even get there and all of that? So this is basically the breakdown of how cells are laid out in a certain, like a skin or whatever. So we have macrophages that are going to eat things up, dendritic cells that's also sort of like eat things up in areas. We have fluid outside of cells out here. The capillaries that are moving lymphatic fluid around and bringing things to and from different areas. Oh, we get a tissue cell of whatever is in it, like the skin cell or whatever, wherever you're looking at. And then the capillaries that are bringing blood, and that means oxygen and everything, nutrients and everything like that to our cells. And then we have connective tissue. Now, what do I mean by this says reticular connective tissue? So that's all these like blue lines. So it's stuff that's kind of holding everything together and creating like a framework for whatever the organ is or whatever it is. Okay. So it's important to understand all of this because cells will have to move in between all of this. Okay. We talked about phagocytes earlier. I don't want to talk about this. Phagocyte being, this is phago being eat. And cyte being cells, mononuclear, meaning one nucleus, phagocytic cells. Okay, so we have areas that are vulnerable to infection, and these will be areas that will kind of drain to any of the immune system tissues to help protect themselves. So essentially what's going on there is like these areas down here is what we're talking about. So we have the skin-associated lymphoid tissue. This is supposed to be S. The mucosa-associated, the gut-associated, and the bronchus-associated. So you can kind of get an idea of where these are located, but these are lymphoid tissues in those areas because these are vulnerable areas that need to be protected a little bit better. So now we have more lymph tissue there to protect those areas. That's the idea. Okay, the circulatory system and the lymphatic system are very, very closely linked to one another. You know, that's pretty much all I kind of want to say about that. They're linked to one another. Well, they're literally like quite literally linked, but I mean, like, what I'm trying to say is they operate along with one another, even though they have different functions. They are literally linked, of course, because that's kind of how the lymphatic fluid is going to drain, you know, to different areas and stuff like that. But and essentially, a lot of the movement of your lymph fluid is going to be from movement, like in your body. And it does hook up back with your circulatory system, but it's not necessarily pumped by your heart. It's just because of the way that it is set up here. So the heart is going to be pumping things out in the blood and all of this to different areas and capillaries and all that. And then these are veins, the blue, coming back. However, also coming back with it will be lymphatic fluid. So see, the lymphatic fluid is only moving in one direction and hooks back up with the circulatory system, which will push it together with the blood back out to the different parts of the body and then come back out as you know, lymphatic fluid and get moved around kind of that way. But you get this moving like with your movement of your body. There's no heart pumping that. So that's what I want to be clear about. Okay. Lymph is plasma-like fluid carried by the lymphatic system. It's just the fluid that all your white cells are going to be moving in with. It's got proteins and salts and whatever, you know, your cells, those white cells might need to be happy. It transports the white cells. It also transports fats and cellular debris that from dead cells and, you know, infectious agents that could have gotten into different tissue spaces and are moving around, getting involved with that. The differences between blood and lymph vessels: lymph moves in one direction only from the extremities towards the heart, and lymph is transported only through the contraction of skeletal muscles, which I was saying is how you move, and it moves the lymph. So if you don't get up and move, or you aren't able to move, or it gets clogged somehow, you can develop edema, which is a collection of the fluid. And it's literally going to be lymph fluid that is collecting there because you're not circulating it anymore. So that can be due to blockages or, you know, immobile limbs. The thymus is an organ where T cell maturation occurs. So what are T cells? They're a special kind of white cell that we're going to get into in the next chapter. And so anyways, it's a triangular region in the pharyngeal region, which is like up at the top up here. It's largest mostly whenever we are born, and then it just gets smaller and smaller as we get older, essentially, because we don't need it because we've essentially matured the T cells that we need. That's the idea. Anyways, I don't think, you know, they sometimes say that they don't think that, you know, people should live as long as we have. So that's the idea. That's why we're outliving our thymus or whatever, but I don't know, whatever. You know what it is, okay? It's for T cell maturation. Lymph nodes, part of the lymph system. Lymph nodes are like bean-shaped organs. I mean, like they are like if you took a pea pod, a normal pea pod, not like edamame, smaller than that, like an actual like sweet, no, not like peas, but yeah, sometimes like peas, a little bit smaller than regular peas. I'm trying to describe the shape and size. They are shaped like beans. And they are smaller than peas. I would say smaller than maybe half a pea, typically. If you're having an infection, they can be bigger. They're not usually as big as peas. That's usually pretty large. So, but yeah, they're pretty small. But yeah, They're bean-shaped organs. They are in clusters along the lymph channels, and they have a lot of aggregation in the armpit, groin, and neck. And we have quite a few in those like galt, malt, salt areas. So these are for filtering the lymph out, getting like the white cells out to come interact and do what they need to be doing in the lymph node itself. Typically, this is where like B cell interaction is going to be going on as well. So we'll get into what B cells are too in the next chapter. The spleen is also considered part of the lymphatic system. It's in the upper left portion of the abdomen, and it filters blood, and filters out like dead, red blood cells and gets rid of those for recycling and all that. It does have an immunological function as sort of like a lymphatic tissue, sort of like the lymph nodes do. Doesn't really necessarily work quite as specifically as lymph nodes do as far as, antibody production location, B cell, interaction and all that. But it does filter pathogens from blood for phagocytosis by macrophages, which we'll get into what those are soon. Don't worry. Whole blood itself, we're talking about whole blood. It's the blood cells, the red blood cells, actual cells, the plasma, which is the fluid portion of your blood. Any of the fluid non-cellular portion of the blood is called plasma. And then if we ever talk about serum, you'll be talking about serum a lot in like the hospitals and stuff like that. And your lab will probably yell at you if you are somebody that works with the lab. They will call you down and say that, no, we need a serum tube. We don't need a plasma tube, or we need a plasma tube. We don't need a serum tube. And you're going to have to know what that means. In serum, we're talking about it's plasma, but we don't have any clotting factors in it. So that's going to be for immune testing and immune therapy and stuff like that. So sometimes you want to keep everything so you can get layers and everything like that. So there's a better picture over here. So if you're wanting plasma, you want a plasma tube, typically you'll get a nice, good, clean separation like this, and you get these layers when you spin them in a centrifuge. And you get this layer called a buffy coat, and it's like a white coat on top of the red blood cells. That is the white blood cells. That's what buffy coat is. It's all the white blood cells in your blood. That's it. And then in that sample, serum essentially is just like if you let it clot on its own, typically not adding anything to it. So just letting the blood sit, and then you form these kind of ugly clots at the bottom that are a little bit nastier, but yeah. It's kind of the difference there. So a lot of these tubes, I'm not going to go through the colors of the tubes out here, but a lot of these, if they have additives in it when we're talking about plasma, typically, if they have additives, tiger top, red tops usually are serum. I think some gold tops are too, but I don't know. I don't really remember those. All right. Right. This is the cells, man. I don't know. There's a lot of cells involved in the immune system. You start out with a bone marrow stem cell. It's a hematopoietic stem cell. That is an important term to be. You don't necessarily have to write this term out or anything. Be familiar with it. If I talk about it later, you'll know what I'm talking about. If I say hematopoietic stem cell, it's the precursor for all of the white cells. So we have the stem cell, that it can become a stem cell for blood cells, for everything that's not a lymphocyte, or it can become a lymphocyte stem cell. So if we go lymphocyte stem cell, it goes through some differentiations that I don't need you to worry about, but essentially it will become a T cell or a B cell. Those are our lymphocytes. Those are the lymphocytes. Those are the lymphocytes. None of these other ones. These are lymphocytes. Don't even try to bring in a macrophage. That's not a lymphocyte. It's a leukocyte. Okay, anyways, the non-lymphocytes over here, okay? We can now differentiate this precursor out to where we can get platelets. They're not cells. They're like parts of cells involved in clotting and inflammation. Then we can get red blood cells, right? We can also get what we're concerned about here in this chapter, our granulocytes. And our granulocytes are our neutrophils, our basophils, and our eosinophils. These are important to know that these are each something that exists and kind of what they are.
definitely neutrophils. Those are the most common white cells in your entire blood. And then we also have the agranulocytes, the monocytes. But notice that even though it is also a granulocyte, which means not granules, you can see it in the picture. This is what they look like when they stain. You see those little dots in there. Those are what they're calling granulocyte, like grainy granules. Okay, you see how the monocytes, the T cells and the B cells don't have that. They are agranulocytes. That's just based on image. Okay. Whenever you look at them under the microscope, literally, that's how they differentiated those. So monocytes are like general phagocytic cells that can eat up bad guys. And essentially, they're sort of like a general use kind of thing. And they can break up into macrophages, which are like our big old fighter, like I don't say big old fighter, but like they're phagocytes that are going to be very specific. And then we have dendritic cells that are kind of the same idea as the macrophages, but they're in specific tissues. And we have certain kinds of macrophages that'll be in their specific tissues as well. So they're just breaking down specific kinds of monocytes, which are just phagocytes to eat bad guys. That's the idea there. So these are kind of their own like group here. These are your phagocytes that are your typical eat up a bad guy sentry guys. So those are your typical sentries. We do have the granulocytes doing that as well, but they do it in a different way, kind of. They have their granules. So let's break it down. Let's go through this. White blood cells. These are all white blood cells all together are known as leukocytes, okay? We stain them and look them under a microscope and see what they look like, and that's how we've named them. That's literally how we've named them. Granulocytes, agranulocytes. Neutrophils are the first ones that we're going to talk about. Neutrophils are 55 to 90% of all of your circulating leukocytes, your white cells. These are the most numerous. They produce toxic chemicals, and they are involved in phagocytosis. So it's eating up bad guys. These are probably the most, I mean, they're most numerous, definitely going to be out there doing like a lot of the work. All right, eosinophils. These are also granulocytes. I'm not too worried about like knowing like what they produce, but they do have inflammatory chemicals. They typically are associated with protecting us from parasites. So help kill, will coat parasites in antibody, and then these guys will come along and help destroy the parasites with the stuff they carry. Okay, so there's usually large eukaryotic pathogens like worms, stuff like that. Basophils, they seem to release histamines. They make up a little bit of it all, but we don't really know for sure what their specific functions are. So I'm not too concerned about these, but I guess I would be aware that they do release histamines. Other things as well, but so the agranulocytes, these are the ones that, you know, they don't have the granules. They have a globular non-lobed nucleus. It's big old nucleus in the middle of it. And they have two kinds. Like what we were saying, I kind of divided it up into the lymphocytes, which is the B and T cells. That's one kind. And then the monocytes. Remember the monocytes? The monocyte is the phagocyte that's going to, you know, eventually differentiate down into macrophage, which we rated as this, and macrophages and dendritic cells. So monocytes are the largest of all of the white blood cells. They comprise 3 to 7% of the white blood cells in circulation. The cytoplasm of the monocytes holds granules. They are there. We can't see them, okay, but they are there. That's why they're agranulocytes, just FYI, I know. But they have stuff to digest bad guys. So they hang out in the bone marrow and they get discharged into the bloodstream as they're needed. And to keep up sentry and all of that. Macrophages are monocytes that left blood circulation and went to the tissues.
So these ones circulate. These ones are in the tissues. They can multiply themselves. They can live for a long time. Yeah. So I don't know. Macrophages are like super important in a lot of this, but we don't really get into like a lot of details with them. Lymphocytes. Really, that's all. We're not even going to talk about dendritic cells. Okay. Lymphocytes are going to be 20 to 35% of all of your circulating white blood cells, your leukocytes. So lymphocytes are leukocytes. It's estimated that about 1/10 of all adult body cells are lymphocytes, though. So I don't know how that makes any sense, like, but whatever. I'm not gonna get into it. So we're only going to be exceeded by red blood cells and fibroblasts as far as, you know, how many of a certain kind of cell we have in our body. Okay, lymphocytes. We have 3 types: B cell, T cell, null cells. Don't worry about the null cells. I'm never going to ask you about them. Never. So definitely no, as far as you're concerned, just B cells and T cells. B in the B cell stands for bursal equivalent. The bursal equivalent is like a bursa is a sac in like birds that they studied. They did all this research on, and then they realized that we don't have one. They're like, oh, okay. These are like that. Okay, we get it now. So that's their bursal equivalent cell. So we just call them B cells. T cells are thymus derived. We talked about the thymus already. It's that organ. It's like here where T cells mature. So, okay, phagocytosis. All of the phagocytes are listed here. Definitely be able to list them. Neutrophils, monocytes, macrophages, mast cells, dendritic cells. So you should definitely easy get these first 3. And then the other 2 you might have to pull out of your butt in some way, but like, I don't know, the dendritic cells I always remember, but like nobody else does. So neutrophils, monocytes, the macrophages, and then we have the mast cells, and then we have the dendritic cells. So, I mean, remember those 5. Those are our immune cell related phagocytes. So what are they going to do? They survey the tissue compartments. They look for microbes that aren't supposed to be there, particulate matter, junk that's not supposed to be there, and then injured and dead cells to break those down and get rid of those. So they're just kind of cleanup crew, right? Sentry and cleanup crew. So they break these down, they get rid of them, and then they also are able to recognize things as being kind of foreign, if you will, and not non-self. We'll go back to that concept again. And then they can alert the immune system that, hey, we got something that we need to take a further look at. Okay. Oh, I was close. Pathogen-associated molecular patterns. I'm not going to make you guys write this out, but it is a good thing to at least know what these are. Okay, so I'm mostly going to call them PAMPs. I'm not really going to break them down into pathogen-associated molecular patterns, which I was trying to say microbial, whatever, but it doesn't really matter. These are things that the phagocytes, the important thing that you know about a PAMP is what it does, right? It's recognized by phagocytes and other defensive cells as automatic no-go. This is a bad guy. Serves as a signal that this is not something we have in our cells. So anything that is like that triggers our cells to be like, this doesn't go here. Peptidoglycan, our cells should not have peptidoglycan, right? So that's something that is a PAMP. And we also have something called lipopolysaccharides that are found in bacteria. And this includes something associated with that endotoxin that we've talked about previously. Remember what endotoxin is? You should be thinking, and when you hear endotoxin, gram-negative bacteria, right? Only gram-negative bacteria have that. And then double-stranded RNA that's found in viruses. So we shouldn't have double-stranded RNA structures. Okay. This is how phagocytosis works, and you would want to know these steps. The first step being chemotaxis. Chemotaxis means that that cell was called there by molecules in the area signaling it to move in that direction. Okay, chemotaxis. Taxi, literally like moving. Yeah. And then chemicals in the area, haul it over. That's chemotaxis. That phagocyte comes over and then it's moved into the area. So this is some bacteria. See how tiny they are compared to the cell? This is what I'm trying to say. They're not anywhere near the same size. Not that this is like to scale, but they are covered in PAMPs, right? So like lipopolysaccharide or peptidoglycan. So here our body, our cell, our phagocyte is going to recognize that as bad once it interacts with it. So essentially, we have adhesion of the bacteria. The bacteria with the PAMP on it is going to stick on to the surface. Then it's going to engulf it in because it interacted with surface stuff, those recognition receptors that saw that PAMP. These receptors saw that PAMP and took it in. So it's taking it in. And then we got the phagosome, which is the vesicle that's formed by eating it up, encircling it. Then we got the phagolysosome. That is the phagosome that we made from eating it up. And then a lysosome comes in and starts melding with it. That lysosome has stuff to break it down, breaks down the peptidoglycan, right? And then we have killing and destruction of the bacterial cells as a result of all of that. And then we will release that debris because we don't need it, or we can use it for whatever parts in the cell or whatever. So we can reuse stuff. It's okay. So that's how that works. Don't definitely know those steps. I would shorten it down. I would probably say, chemotaxis. And then I would say 2, adhere. Then I would say 3, engulf. And then 4, I would just say vesicle. I mean, it is phagolysosome. I just don't want to write it right now. 5 is combine with lysosome. And then 6 is kill. And then 7 is release debris. So you can write these out. I would know these steps for sure. Okay. So you understand what's happening stepwise when we're talking about this, because we're not done talking about it. So monocytes are like the ones that were circulating, right? So they're going to be transported into macrophages, transformed into macrophages after they get out of the bloodstream and into the tissues due to chemical stimuli. So that's chemotaxis. So something in the tissue has called those macrophages, those monocytes, into those tissues to become macrophages and come check things out. So histiocytes are a certain kind of macrophage that go to a certain tissue and remain there always. So they're always associated with that tissue. Certain kinds of histiocytes include alveolar macrophages, literally macrophages that hang out in the alveoli of your lungs, Kupffer cells in the liver, dendritic cells in the skin, macrophages in the spleen, lymph node, bone marrow, kidney, bone, and brain. There have specific ones for those areas, and they just stay in those areas and they don't move out of it. It's histiocytes. Okay. Dendritic cells themselves have long little, they say long thin processes, like little extensions coming off of the cells. They move from the blood into, I don't know, into the, I'm just going to go ahead and go on with that, lymphatic tissue where they trap pathogens. Yeah, I don't know. They basically do a lot of the same stuff like a macrophage would do, but they're going to be in like skin cells and stuff like that. So not skin cells, sorry, the skin area tissue in general. Yeah. They can also hang out in the lymph nodes and the spleen and serve as antigen presenting cells there, which means that they are going to take like molecules that they think might be bad guy molecules and, presenting them to cells to see if they think we should react, how we should react. It's like reconnaissance. Okay. So we have erythrocytes, those are red cells, and we have platelets that are going to be involved in clotting and inflammation. Good. When we have inflammation, we have these 4 kind of signs and symptoms, okay? Rubor for the redness because of the increased circulation there. So more blood is in the air, so it turns red. calor for the warmth, because blood is warm. So the more blood there is there, which we already see for the redness, the warmer it's going to be, right? Tumor, which is the swelling. That's essentially fluid escaping from the tissues itself due to the damage in the area. And then dolor, which is the pain, because that's literal stimulation of the nerve endings whenever we do injure ourselves. So know what these are. Okay, mediators of inflammation and immunity. Okay, I'm not going to make you guys know each of these specifically. I would definitely be familiar with the fact that tumor necrosis factor, or TNF alpha, that interferons, that interleukins, doesn't have to be 1 or 6 or whatever, just interleukins in general, that these mediate inflammation and immunity. I need you to know all this other, okay. And then as we go down, histamine and serotonin and bradykinin, but I mean, for our purpose, I'm not going to get too much into like serotonin's, purpose in this, but like that these are vasoactive mediators. And that's these ones down here regulate lymphocyte growth and activation, but I'm not really going to be asking you guys about these. Okay. Yeah, there they are though. I told you about them. Okay. White blood cells can squeeze in between these, intercalate, if you will, between other cells via, they get called to an area by chemotaxis due to these chemotactic factors. Believe it or not, that's what they're called. They move into that tissue space from the blood between the epithelial cells, endothelial cells of the blood vessels. By something called diapedesis. Okay, I'm not going to ask you guys too much questions about the diapedesis, but they literally are just squeezing in between those spaces in order to get to that area that they're being called to, if you will. So fevers, what are fevers and why are fevers? Fevers are abnormally elevated body temperatures. It's a universal sign of infection, typically. Other things can cause it as well, cancer, organic illnesses, allergies and stuff, they can also cause fever. So essentially all fevers are caused by a pyrogen. That's a term to be familiar with. Releases prostaglandin, triggers your body to release prostaglandin to dysregulate the hypothalamus, whatever the pyrogen is. The pyrogen could be endotoxin, for example, or it could be something else associated with a pathogen or a toxin or something like that. That just means anything that causes a fever. It also leads to, so we have high temperature, increases, which increases the metabolism and the cell activity in the area, and it decreases microbial growth rates. So that's something else that it does. And then it also causes the release of transferrin. So that is less iron available for microbes. So these are all good things that fevers do for us. We consider low-grade fevers to be anywhere from 100 to 101 degrees Fahrenheit, moderate to be 102 to 103, and high to be 104 to 106. Of course, always consult your doctor whenever you are considering whether or not you should take, anti-pyrogen type medications to lower your fever, whether you want to lower your fever at all. I am one of those people personally who keeps my fever around because it's there for a reason. But if it gets to a certain point, usually 103 for me, then if it gets above 103, then I will take something for it. But you need to be, consulting your doctor on making those decisions, and definitely don't necessarily apply these things to children. Consult their doctors. So pyrogens can be exogenous from outside of the body. They can be produced by infectious agents like viruses, bacteria, protozoans, and fungi. It can include endotoxin, and it can be also include blood products, vaccines, and anything that came from outside of our body, exogenous. The endogenous versions of things that can cause fevers within us, came from within us, is basically any triggers that are being released by our phagocytes. They can signal our body to make a fever for whatever reason. They might see a PAMP of some kind. There might be something else signaling them to make it, but essentially these signals will lead to a fever. So interleukin-1 and tumor necrosis factor, those cytokines that we were talking about earlier that cause signals to the cell. Cytokine is essentially just a term for molecules that cells use to communicate with one another. So interleukin, interferon, tumor necrosis factor, those ones that we talked about in that previous slide before. I told you I was going to use these terms. I'm just going to start throwing them in there, and I'm not going to explain them very much anymore. You don't need to know exactly what they do, just know that they are communicating molecules to allow cells to communicate. Okay, so apparently we're going to just get right into these. I didn't realize it was the next friggin' slide. Interferon is a small protein produced by white blood cells and some other cells as well. And usually it's used for treating viral infections, so things that are inside the cell. And it's involved in immune regulation and communication as well. Essentially the way that it would work in a viral infection, interferon, okay, so we got a virus coming in. Virus comes in, we got a virus infection, we release our viral nucleic acid into the thing, and we're going to start making parts of our virus. When this is going on, our cell knows because it's using all of our cell's equipment, right, to do all this stuff. So it interacts this interferon gene. It activates this interferon gene. So as it is activating that gene, it'll make interferon, and our cells will start to secrete it. And then cells that are nearby can receive it as a signal. So that whenever the viruses that are being made by this cell that's signaling, or other cells that are nearby that have it, or just viruses in the area, come in to interact with these receptors and come in to infect the cell, these cells have already been activated, and now they can start degrading and blocking and making it so that we can have antiviral proteins and all that, and protect the cells nearby. So it's like a mayday signal, or it's like more like a warning signal, like, hey, there's an iceberg ahead or whatever. Not quite like an iceberg, I guess. There's bad guys. We got pirates on our ship. Get ready is what they're saying. And so the pirates come on over, and that ship was ready. So that's what interferon is like. It's like sending a secret message to tell the other cell that the pirates are coming. Complement is one of my favorite things in the innate immune system. It's also very important in the adaptive immune system, the selective one. We'll get into it, don't worry. Complement, it consists of about 30 different kinds of blood-based proteins. I don't need you to know that. I just want you to know that this is called complement and that it works to destroy bacteria and viruses. Okay, does this, it's called this, does that. It causes a cascade reaction. And that's why I love this. I just think it's so interesting. That just means that, like, essentially, we get one protein that signals, breaking down another protein. And when that protein is broken down, it can, signal another protein. And if this other one got triggered, they'll combine together and then they'll signal. It's like they're all like dependent on one another. And eventually at the bottom of all this whole slew of proteins, we can signal to do certain things. So yes, the first substance in a chemical series activates the next and so on until the desired end product. It's a cascade. This is a depiction of complement pathway. This is the classical complement pathway activation. Classical complement pathway is dictated by antibody. Here we've got antibody. This complement protein here interacts with the antibody and signals for activating C2 and C4. When they are activated, they can break apart and some of their parts come together, whatever. They make C3, whatever, it breaks apart. And then what happens? This is the whole point of everything. The antibody signals, this is the take-home message. Antibody signals the complement cascade that can lead to opsonization. I know that's a word you don't know what it means, and we will know it in a minute. I'm going to drill it into your brains. Cytolysis, which is breaking down cells. We've had that word before. And then inflammation. We just learned about what that is. Okay. that's the idea. I don't need you to know like C1 and all that. This is the alternative pathway. Essentially, you see how there's not antibody here. So we have, these factors here come interact with a lipid carbohydrate complex on the outside of a microbe. Essentially, that could be what we call that lipopolysaccharide or endotoxin is a type of this. It's not only endotoxin, but endotoxin falls into that. Anyways, it signals these complement cascade. And guess what we get? Opsonization or cytolysis and or inflammation. You can get all three. Then we have the lectin pathway. It's the same idea, right? Instead of recognizing microbes lipopolysaccharide or microbes that are coated in antibody, in this case, we have proteins that come and interact with carbohydrates on the surface of the cells that have mannose. Lectin is a protein that interacts with that. So lectin interacts with the mannose and triggers the complement cascade. Same results. Very useful. It's pretty much how the big time is controlled in your body when we're fighting off bad guys. All right. So this is the big idea of what's important about how the complement cascade works. When I talk to you guys about how you can trigger opsonization, opsonization is enhancement of phagocytosis with coating. I don't need you to know that it's 3B. Opsonization is enhancement of phagocytosis, eating up the cell by coating it with something. You can coat it with proteins from complement. You can also coat bacteria with antibody, and that helps with phagocytosis as well. So that's also called opsonization. Okay. So those are both opsonization in that case. So that's one way where our complement's going to do its thing. Another way is triggering mast cells that release histamine to lead to inflammation. Inflammation is going to call more cells in there to start fighting more battle, attracting more phagocytes to help engage the whole immune process as a whole, and to really get in there. And so that's why we have blood flow there, and heat and swelling, because all that stuff, it has to go to that location in order to start with that call to action, if you will. So the mast cells will go out and do that. That's also increased by action by the complement cascade. Okay, so we've got opsonization. That's our number one. Number two was that inflammation. We just learned a new word, opsonization. You're not going to remember it. And then number three, we have formation of these channels. The complement cascade proteins come together and literally form holes into the cells, like membrane, transmembrane channel. It goes through the entire membrane of that bad guy cell, the target cell, the bacterial cell, whatever it is, and then it bursts it open through forming those channels. Everything just spills out. So that's what cytolysis is. Cytolysis is cytolysis, is breaking open cells. We do that by the formation of what they call a membrane complex. So I would remember that as being the complement triggered membrane complex. Okay, that's what complement does. It's really important in our immune systems. Because I'm sure you guys know that we have immune systems and they fight things off. This is me telling you how it does that. So we talked about the first line of defense as physical barriers and chemical barriers. We talked about immune surveillance, which is a lot of our phagocytes kind of hanging out, going around, checking out things, eating things up, and then reacting to PAMPs, right? Pathogen associated molecular patterns. Those molecular patterns, I was trying to remember it, but no PAMPs for sure. I'm not going to make you write out what PAMP is. I can't remember it. I can't remember it. How can I ask you guys to? Right? That's only fair. Immune surveillance. Okay, so then blood cell types. We talked about mostly the white blood cells and what the different ones were and what they do. And then adjuncts to the immune system. So things that are going to help the immune system out and help it actually function. We talked about the importance of inflammation. We talked about fever and what it does for us. And we talked about the complement cascade. very important, the complement cascade. So definitely read up on that one. That's one that you're going to want to be aware of, those 3 things that the complement cascade does for our cells. It's important stuff. And I'll see you guys next time for chapter 15, I believe. We're on 15, yeah, 15.